When a patient in an acute psychiatric crisis arrives at the emergency department, the environment is inherently chaotic. Clinicians must make immediate triage decisions. In that high-stress window, emergency staff face a severe diagnostic limitation. Distinguishing between acute schizophrenia and bipolar I disorder currently relies entirely on subjective longitudinal observation. There is no rapid biological test to tell the two apart. This ambiguity has severe clinical consequences. High initial misdiagnosis rates mean patients are frequently subjected to a 1 to 3 year trial and error protocol of generic antipsychotics, delaying access to targeted interventions like clozapine. Yet, the biological markers to differentiate these conditions exist. The Sentinel principle demonstrates that peripheral venous blood can serve as an accurate transcriptomic liquid biopsy for the central nervous system, capturing a distinct mRNA expression signature for severe mental illness. The ongoing reliance on subjective clinical observation is a structural bottleneck preventing rapid precision intervention for these patients. To solve this, we are engineering the SZ meter, a desktop, point-of-care microfluidic diagnostic analyzer designed specifically for psychiatric triage. The underlying biological science is already established. The instrument licenses the VOTR test, a highly accurate 18-gene mRNA expression panel that has been thoroughly validated in central laboratory studies to separate schizophrenia, bipolar, and control cohorts. Moving this quantitative assay into the emergency department turns a hardware miniaturization task into a complex problem of data architecture. The core engineering innovation of the SZ meter is its dual-stage fusion architecture. This system combines rigidly locked biological data from the blood panel with dynamically updatable observational data from the patient encounter. The design goal is to build an instrument that satisfies rigid regulatory oversight while operating within the fluid, nuanced reality of the clinic. The first stage of the biological pipeline is executing complex sample preparation on a disposable microfluidic cartridge. The hardware must automate solid-phase RNA extraction directly from a 3 milliliter draw of whole venous blood. Inside the cartridge, the system initiates guanidinium lysis to break down the cells and inactivate enzymes, followed by solid-phase extraction across magnetic silica beads. This specific sequence is required to wash away proteins, genomic DNA, and inhibitors like heme that would otherwise corrupt the read. Next, the system performs an indispensable reverse transcription step. Human blood cell RNA is highly unstable and single-stranded. Reverse transcription converts it into stable, double-stranded complementary DNA, which is the only format the subsequent amplification stage can process. The liquid cDNA is then spatially multiplexed. Because current optics cannot resolve 18 distinct chemical signals in a single fluid droplet, the sample is allocated across discrete target wells, preloaded with dry stabilized primers and fluorescent probes. This rigorous physical and chemical processing is the absolute prerequisite for translating volatile biological molecules into a readable quantitative signal. Unlike qualitative viral swab tests that merely detect the presence or absence of a pathogen, the SC meter must calculate the exact relative abundances of 18 different low-level human transcripts. This relies on real-time PCR, an exponential process where target templates amplify by a factor of 2 to the 35th power over 35 thermal cycles. This graph illustrates how sensitive that amplification is. It compares a perfect PCR amplification curve against one distorted by just a 5% thermal drift, showing how the error compounding geometrically over 35 cycles. If the temperature is off by even a fraction of a degree early in the cycling phase, the exact relative abundance estimates become useless noise, which would trigger a false diagnostic output. To prevent this exponential drift, the hardware architecture uses a Peltier block or a microheater array designed to maintain strict spatial uniformity of plus or minus 0.1 degrees Celsius across the entire reaction array. Ultra-tight physical hardware control is the only way to guarantee the biological data entering the diagnostic algorithm is mathematically trustworthy. Once the amplification completes, the firmware tracks the raw optical fluorescence, normalizes it against an endogenous reference, and converts it into a distinct expression vector. This 18-gene expression vector is then fed into a stored biological classifier, a logistic regression algorithm that maps the normalized data into a specific diagnostic probability. To achieve an FDA de novo classification, the coefficients of this mathematical model must be completely frozen and unchangeable. However, a locked mRNA model creates a context gap. It cannot perceive real-time behavioral variables that bear heavily on psychiatric triage, such as severe agitation, mood swings, or extreme sleep deprivation. If developers tried to open up the biological algorithm to absorb these evolving clinical and behavioral norms, it would trigger a multi-million-dollar clinical revalidation of the entire FDA-cleared assay. Relying on a single, locked biological algorithm is incompatible with the fluid, context-dependent nature of psychiatric triage. The SC meter solves this through its architecture. It establishes a separate secondary data pipeline that runs completely parallel to the biological one. This diagram shows the dual-lane data architecture. The bottom lane securely processes the biological expression vector through its locked FDA-cleared classifier. The top lane simultaneously captures the observational feature vector. Concurrent with the 60-minute blood processing, the instrument's touchscreen administers a structured symptom assessment, while passive sensors like a camera or microphone capture behavioral cues. This top lane produces an independent observational feature vector, capturing the exact human context that the locked blood panel cannot see. A second, independently locked classifier then merges them, fusing the observational vector with the biological probabilities to produce a single combined score. Because this secondary AI fusion layer is versioned independently, it can be recalibrated to new clinical norms on its own regulated cycle without disturbing the locked biological FDA clearance. This dual classifier architecture successfully bypasses the regulatory bottleneck, granting clinicians the certainty of hard biology paired with contextual reality. The final computational output from the fusion classifier is a single combined probability score, indicating whether the patient matches the profile for schizophrenia, bipolar 1, or a normal control baseline. To make this complex, multi-vector data immediately actionable for non-specialist clinical staff, the system condenses the output into this graphical user interface. The result is redundantly encoded. The pointer's position on a 0 to 100% scale shows the probability, while the color hue at that position corresponds to the specific psychiatric class, ensuring rapid, unambiguous comprehension in high-stress environments. The interface intentionally retains a secondary background indicator, displaying the biologically only probability, ensuring full traceability and allowing the clinician to audit the machine's logic. The interface serves as a translation layer, hiding intense mathematical and physical complexity behind a simple, actionable triage directive. Engineering an accurate microfluidic machine is a means to an end, materially altering the patient's clinical trajectory. Providing objective, day-one diagnostic certainty allows emergency clinicians to confidently route high-risk patients to aggressive therapeutics, like clozapine, at the first onset of disease. This immediate, targeted intervention replaces the historical cycle of misdiagnosis and steady neurodegenerative progression. The hardware is the necessary physical infrastructure to deploy precision psychiatry at scale. True precision medicine in psychiatry requires an architecture capable of recognizing exactly where rigid biology ends and human context begins.